Okay, hello everyone. My name is Qian Zhao. I'm an earth scientist here at EMSL. So for MONet program, I am part of the data generation team. I oversee the different data types that generated throughout our lab and also the JGI and the Neon network as well. So here, I think everyone here for our MONet database. So data is the center. So I will go to the full list of the data set that we provided through MONet program. So here is a table showing you all the data types that we provided. You can find this on our MONet website as well. But we have more than 30 different data types included into this database. So as John mentioned, we group them into different categories. The first one is the biogeochemistry analysis. So we have this gravimetric water content, microbial biomass, carbon nitrogen measurement. pH is a common measurement for all the samples, soil samples, and inorganic nitrogen, phosphorus measurement by extractions. And also some other cations that help with this geochemistry understanding of your soil. And of course, the cation exchange capacity measurement as well. So for microbiology, we also provide a potential enzyme activity measurement, specifically for beta glucosidase activity that measured by colorimetric assay. And we have the soil respiration that measured, and as well as the total carbon and nitrogen in bulk soil. So again, this biogeochemistry analysis is common in all different networks or database, but we want to have this standardized protocol that measure across the board so we can compare different samples from different locations. The second category is the physical property analysis. That include the soil pore network structure measured by XCT, and the soil texture. We provide you the percentage of sand, silt, clay, and measured by hydrometer. The third one is the molecular analysis. Here we have this FT-ICR, high resolution mass spec, to measure the water extractable organic matter that represent the label carbon pool. We also use FT-ICR to measure the molecular composition of the mineral associated organic matter pool that represents stable carbon pool in soil. In partnership with JGI, we have this metagenomic sequencing done on this in this database that provides you the molecular, the microbial community composition as well as the structure. As John mentioned, we have these three primary analysis that provided by MONet. They are XCT, give you the microstructure of the soil cores, and FT-ICR, give you the high resolution molecular composition of your organic matter pools, as well as the metagenomic sequencing. So for the rest of my talk, I'm going to dive a little bit deeper into those three analyses to give you an overview of the workflow of those three analyses. So first is the metagenomic sequencing in partnership with Joint Genome Institute, JGI, to conduct this analysis. But we do the DNA extraction in-house here at EMSL. Isabel is leading that effort to do the DNA extraction. Here we use Zymo research kit to do the extraction and follow their manufacturing protocol step by step. Once we got this DNA extracted, we quantify on nanodrop first. And once those quality been passed, so we pass those samples to JGI for sequencing. And here I give a shout out to Satish, who oversees this metagenomic sequencing protocol of workflow from MONet side of view, and Yung Song help with the bioinformatic analysis. Once we send sample to JGI, they first do the QC check on those samples and do library preparation and run the sequencing and do the QC check again once they got the raw data. So after the raw data, they can do the sample, the data processing through assembly, read mapping, binning, as well as the functional annotation. So all those key steps, key analysis can be done in their data portal. They have this integrated microbial genomes and microbiomes platform that can process the assembling, binning annotation. But both the raw data and processed data are going to be available from MONet data portal, so you can access to them. Right now, they are, for this first year of MONet samples, they have been analyzing. So we implement this pipeline, and it will be available to you soon. But if you want to understand how those JGI platform provide you those data analysis, you can go to their published paper, this JGI metagenome workflow. I think to me, it's as a no background in metagenomic sequencing, this is really helpful to me, and it's very clear to understand how they process data step by step. But also, there are a few links down here at the bottom. They provide you the workshop webinar series from past years. So you can go there to understand their functions and the tools that provided by their data portal. If you are interested to see how to use this metagenomics data for answering your research question, you can also attend their annual meeting. There will be some user to present a research project by using the metagenomics sequencing data. And we also have a colleague here from JGI. If you have specific questions, you can reach out to any of us to follow up on some specific questions. With that, the next analysis is the XCT, X-ray computed tomography. So it provides you the soil pore networks, network structure. So we have Thomas and Anil here is leading that analysis in EMSL. XCT is a non-destructive X-ray imaging tools to examine or visualize your soil cores of those the microstructure in your soil cores. And here, this image on the upper left is our XCT instrument here at EMSL. It's hard to see the resolution; it's not clear. But we have this MONet, one of the MONet soil cores in here to be measured. And based on different density, we can tell the different the spatial difference between the soil aggregates and the pore space. So then we can generate this 3D image, 3D structure of our soil cores. So here I'm showing you a workflow of this XCT analysis. I won't go into details step by step here because Thomas is gonna give you a lecture to talk about the theory and how to generate this data. But I just want to touch on the key steps. So first we do the image acquisition to get those 2D image layer by layer across the z-axis of the soil core. And second, we do the image reconstruction. That means we put those 2D images together to build up this 3D structure of the soil core. A third step is the segmentation. So to separate the soil aggregates from those pore networks, so we can have those two systems side by side. And lastly, once we got those pore networks, we can do quantification to generate this major outputs, give you the quantification information about the porosity, pore volumes, connectivity, pore diameters, permeability, flow rate, and so on. So those data can be further correlated or interpreted with other biogeochemistry data or molecular data that we provide here. Also, you can measure by yourself. And of course, some modeling work can be done on this XCT analysis, such as the separation of those this 3D construction of the pore networks. We can use some modeling to do that. Thomas is going to touch on that. And also we can, so the mic is low. I think that's come from here. I get closer to this computer. So we can also use some modeling work to do interpretation with other data tests with XCT analysis. So the next one is our FT-ICR analysis. That's give you the organic matter molecular composition of a labile carbon pool and as well as the stable carbon pool. So here I'm showing you the major workflow of this analysis from all the way up to environmental design and the soil sampling prepare a sample for the instrument measurement on FTICR. And also, once we generate the raw data, we can do the formula assignment and process this, generate this final data products that can be used for further analysis interpretation. Here, Rio and Nicole gonna give you a lecture, tutorial, as well as a hands-on activity throughout these 2 days to let you familiar with how we generate data, how we process data, and how this data can be visualized and further interpreted. Rosie and Jason are here to help with the instrument measurement to generate those raw data. Another view of this workflow is we have the soil core collected, and we do the extraction by water or 0.5 molar HCl, and then to use a direct injection to run on FTICR instrument. We do the formula assignment. We have those amazing figures that based on these thousands of molecules. And then we can do further visualization, exploring the data to interpret this FTICR data. You can also incorporate it into some statistical analysis to do the correlations and interpretation. Yeah, with that, those are the 3 primary analyses that we provided. And also, we also have some workshops and trainings on other analysis, like biogeochemistry analysis that we're gonna provide this afternoon as well.